Then we'll move on to talk about the part B, which is the structure questions. And we can see that, okay, the photo graph below, which shows a storage organ of a potato plant. So in this case, in fact, it would be the butt of the storage organ. So then definitely, in fact, what type of storage organ is shown, then definitely it would be the stem tuber, okay? So how can you test for the presence of the major food reserve stored in this storage organ? In fact, most of you will write down just simply iodine test. In fact, when we talk about how, then in this case, you need to tell me the procedures. For example, we need to add the iodine solution to a slice of potato tuber. Okay, then in this case, for example, if the starch is present, then in this case, it would be the iodine solutions will turn into uh, brown, sorry, blue black, blue black from brown color. Okay, describe how potato plant from this type of storage organ. So in fact, we need to talk about how the potatoes, okay, it can develop or it can form this type of storage organ. Then many of you just simply write down the of how the potato or the stem tuber will grow or it will form a new individual. So you need to tell me more specifically that maybe the green leaves of the potato plant will carry out photosynthesis to produce food, and then the excess food or the excess nutrient will pass to the underground of the stem. Then in this case, it will form the underground stem, which is the stem tuber. So next, okay, so what happened, okay, so what happened, okay, or describe and explain the change in size of the storage organ as the bulb is grow further. So one is growth because it is consuming the nutrient or the food stored in the potato or the inside the stem tuber. So therefore, the stem tuber, it will become smaller in size, okay, smaller in size. Then this is why, because it will some of the stored food, in fact, it will be used for the growth of the new plant or it will be consumed, etc. Okay, that is because so. Then next is the production of new plants from the storage organ, a kind of asexual or sexual reproduction. So definitely it would be a kind of asexual reproduction. So why? Because based on the based on what we learned or what we have mentioned in the previous multiple choice questions, in fact, you will notice that because there is no any involvement of the fertilizations or of the fusions of gamete. Or the fertilization of gamete. So in this case, it would not be a sexual reproduction. So it would be a asexual reproduction. So then, okay, when we continue, an experiment was conducted to investigate the germinations of the pollen tube from the pollen grain. So the pollen grain from 3 different flowering plants were transferred to the sucrose solution of different concentration and incubated at 23 degrees Celsius for 24 hours. So in this case, name the flower part that it will secrete these sugary solution, or which the pollen tube will germinate. Then in fact, it would be the stigma, okay, the stigma. So with reference to the above results, okay, so state 2 conclusions that you can observe. The first one, in fact, the sucrose solution in general, that, okay, it would have increasing, okay, because we can see that when we increase the sucrose concentration, in fact, both plant P, plant Q, and also plant R, there would be an increase in a successful seed the pollen tube germination rate. So in this case, the first part would be, or the first conclusion can be made, okay? So for sucrose solution, sorry, the concentration of the sucrose solution increase. Then in this case, okay, the successful rate, okay, the success rate of pollen tube the germination would be also increased, okay, in these 3 plants, okay? And some students do mention about one more conclusion would be, even though it is 0 concentration, okay, which is not in the answer, but we do think that it is a correct one. In fact, even though 0% of the sucrose solution, the concentration of the sucrose solution, in fact, okay, there is still successful rate, okay, still have certain level of success rate of pollen tube germination. So we can see that even though we try to minimize to 0% of sucrose solution, then that means it may be completely water. Then in this case, okay, we can see that, okay, in fact, okay, there is still success rate of pollen tube germination. So certain percentage of it still success for the pollen tube to germinate. So another one would be the successful rate is different in these 3 plants at the same sucrose concentration, okay? Or the germination rates of the pollen grain of plant Q, and that is the lowest at all the sucrose concentrations used. So a student thought that the result obtained from using 15% of sucrose solution for plant R was due to an error. So how can he find it out this idea is correct? So we can see that, okay, there is a problem, okay, because there is a drop, okay, there is a drop, okay, a drop on the data, okay. So how can he do? In fact, he can repeat the experiment for several times. So by increasing the times that the pollen grain, that means the experiment, okay? So in fact, this can help us to eliminate some possible error or some abnormal data. And this is why it is important for us to increase the reliability of the result. Okay, so with the pollen grain from plant R to take the average successful rate. Okay, so then this can at the other time, okay, we can also try to make our result, okay, a little bit more consistent, okay, consistent and similar to those in this experiment would refute this idea. Okay, so we can try to make the result a little bit more consistent because we have to do a multiple experiments to indicate that, okay, in fact, our results do got similar, so that maybe, for example, if it really dropped, then in this case, this could be some possible, maybe some other reason or other factor that leading to such drop, okay, for it. And then next, for question number 3, we can see that the photograph below shows a flowers from an avocado plant. So we can see that, okay, it will open, and we can see that, in fact, originally it is enclosed, both anther and also the stigma is enclosed inside the flower. But very special for this type of flower, they will blossom, and somehow the anther and also the stigma will open, okay? So in this case, we will be sure that, because one of the way would be it is by the insect pollination. Insect pollinations. Why? Because we can see that the flowers do have a large petals. Of course, this would not be a really good observable features, but in this case, we can see that one of the only features would be it has the large petals. And apart from that, both anther and also the stigma, in fact, they are located in the inside part, okay, of the flower, inside or the middle of the flowers, okay. So in this case, it is not maybe hanging out, or it is outside the flowers. So what will be the advantages for the reproductive part of the avocado flowers to mature at different times? In fact, if you still remember during the lessons, okay, so by having 2 different interval, then this can help to avoid self-pollination of the avocado plant. Why? Because we can see from this part, okay, in fact, day 1, in fact, maybe it only opened with the mature stigma in the morning, while until to the day 2, it will have the opening of the mature anther. Then in this case, maybe the mature stigma already fertilized. That means already received the pollen grain from the other plant. Then they already fertilized in the ovule. Then in this case, there would be no any self-pollinations. So this can reduce the times or the possibility of having the self-pollinations. And in this case, the genetic variations will be increased. Okay. of the offspring will be increased. So an avocado orchard often has both type A and also type B avocado plant. So what is the purpose for planting it? Because both, okay, planting both type A and also type B, this can increase, okay, the chance that pollinations, cross-pollinations of both type A and also type B of the avocado plant. So in this case, okay, by planting both type of plant, it can increase the chance of cross-pollination. Why? Because you can think of like in this way. Again, okay, during the time that's in day one, in fact, plant A, okay, type A plant, in fact, they will have the mature stigma. While on day two, morning time again, okay, in fact, they will also have the mature anther for the plant B, plant type B. So in this case, as both plants, they may not have the same time for the blossoming. So maybe some will be later, some will be earlier. Then in this case, during the morning time, only type A and also type B plant, they can have the cross-pollination because there will be opening of the mature stigma and also opening of the mature anther for type B plant. So in this case, there would be a cross-pollination, but no any self-pollination because it will close at the afternoon time. So in this case, it will not open. So no any pollination, no any pollination during the afternoon time. So in this case, this can reduce or minimize the self-pollination. But in this case, by having type A and type B, then in this case, these two types will have different, they will have the cross-pollination, and this will increase the genetic variations and when they the mature stigma and also the mature anther open at the same time. So in this case, the flowers can be fertilized and develop into the fruit with a higher genetic variations. And this would be the remaining answer for the part C, okay? So maybe, I hope, okay, maybe these videos can help you to understand about these questions in homework 12, okay? So just simply let me know if you have any other questions. Thank you.